All right, so we're going to look at probability, genetics, and percent deviation, or sometimes known as percent error. All right, so we used a coin for this genetics coin flip lab. The reason we did is because heads would represent one allele and tails would represent another allele. Or in this case, the allele would be big A for heads and little a for tails, or dominant and recessive. All right, so if you flip a coin, you have the options of heads or tails, whereas in genetics, you have the option of the dominant allele or the recessive allele. So here we have a person, a man, who is genotype big A, little a. Now, when this person has a cell go through meiosis, remember meiosis is the process through which sex cells or gametes are made. So if this person is big A, little a, they have two choices. They can make either big A sperm or little a sperm. So we have, through the process of meiosis, four different kinds are made, and they are two big A's and two little a's. All right, so again, looking at that, we have 50% are big A, 50% are little a, and at any given time, there are up to 1 billion sperm cells, and so when you get up to a high number like that, the chances of big A are right around 50%. The chances of little a are right around 50%. So a coin flip is a very good analogy for doing genetics. Right. So if we have a case where we have a male, which is big A and little a, and a female who is big A, little a, now we're looking at, right, we have heads, or tails. This is the part of the coin flip lab where we are flipping two coins. So let's take a look at that. If we have the male, which is going to make two possible kinds of sperm as big A or little a, and the female can make big A or little a eggs, then we put that together in what was called a Punnett square, where the male will either give big A or little a, the female will either give big A or little a, and we're left with a Punnett square that looks like this. What that tells us is that 25% of the offspring could have big A, or heads, 25% could have little a, or tails, and 50% could have big A, little a, or heads, tails. Sorry, my kids are playing in the background. All right, and the last thing that I'm going to talk about in this video is percent deviation, which is, again, also known as percent error. So if you expect 50% to be heads and 50% to be tails when you're flipping a coin, we're going to look at how to calculate percent error. So let's say that my results are 6 heads and 4 tails. Now this is my actual. My expected is 5 heads and 5 tails, right? 50%, 50%. Life at home, right? Life at home. I'm not going to edit this either. All right, likewise, if I flip the coin 50 times and I got 23 heads and 27 tails, I would have expected 25 heads and 25 tails, being 50% and 50%. All right, so looking at our data, if I had 6 heads and 4 tails, we're going to look at how to calculate the percent deviation. All right, so the percent deviation is equal to the sum of the differences divided by the total times 100%. So So when you look at the differences, we have 1 and 1. So it's obviously 1 plus 1. Okay, divided by the total number of flips. In this case, it's 10. And so I have 2 divided by 10 times 100%. Well, 2 divided by 10 is 0.2, and then times 100% is 20%. So that is my percent deviation. All right. Likewise, if I'm looking at my 50 flips, if I have 23 heads and 27 tails, if I have a difference of 2 and 2, I take 2 plus 2 divided by the total, which was 50. So now I have 4 divided by 50, and that comes out to be 0.08, which equates to 8%. So my standard deviation went down after I increased my sample size. And this is a trend that we would expect unless you had happened to have like 5 heads, 5 tails in the first one, and you would have ended up with 0% deviation on the first one. So looking up above, we would have 20% deviation and 8% deviation respectively. The grade 8 10 flips and grade 8 50 flips you will do using the data compiled from your the Google form that we did yesterday.